Okay. So, okay. Uh, so in our previous class, we were discussing about the biosynthesis of terpenoids. Anyway, I will make it a presentation mode. Okay, I will make a presentation mode. I will take a pointer. Okay. Biosynthesis of terpenoids. More and this biosynthesis follows a method or pathway called mevalonic acid. The mevalonic acid can be obtained by the combination of right. So biosynthesis of terpenoids. So this biosynthesis follows a mevalonic acid pathway, and this mevalonic acid is obtained by the combination of acetyl coenzyme A in a nucleophilic substitution manner, more particularly condensation manner. So this you may consider it as a melonic coenzyme A. Okay, acetyl coenzyme A, melonic coenzyme A. So melonic coenzyme A, when it is react with acetyl one more equivalent of acetyl coenzyme A, there will be a formation of hydroxymethyl glutarate diester of coenzyme A. So this on partial hydrolysis, one of the coenzyme A group will go out as a living group and forming a acid monoester coenzyme 1. So this one, what will happen? Undergoes reduction. Okay, more particularly, the reducing agent in anaerobic condition is called NADPH. So this will act as a source of hydrate. H minus will supply over here, and this will go out. So that is how a aldehyde and carboxylic acid is obtained. This is called mevaldic acid. This mevaldic acid further reduced with NADPH. NADPH means H minus. H minus attack this carbonyl carbon, convert into a aldehyde into alcohol. This is only mevalonic acid. and how this mevalonic acid is used in the synthesis of terpenoids, that we will look in our next slide. Okay, so this is a biosynthesis of terpenol, how this terpenol is obtained. Okay, that we will discuss now. Mevalonic acid, this is the one. And this mevalonic acid I'm treating with ATP. ATP will act as a source of phosphoric acid moiety. So that means a hydroxy group is converted into phosphate. And ATP is converted into ADP. ADP is nothing but a adenosine diphosphate. ATP is adenosine triphosphate. So ATP gives one phosphoric acid group to this and convert hydroxy into phosphate ester. ADP itself is converted into ADP. So then once again this mevalonic acid, when it is treated with adenosine triphosphate, so it's converted into pyrophosphate, mevalonic acid pyrophosphate. So this mevalonic acid pyrophosphate undergoes dehydration. How the dehydration? It also undergo decarboxylation. This CO2, it will go out, decarboxylation. This bond pair can come over here and this bond pair can come over here. This will go out. This OH and H will go out as a water, dehydration, and this CO2 will go out as a decarboxylation. So that is how a key intermediate is formed. And you know that this key intermediate is nothing but you can consider that 1 2 3 4, okay, 5. Okay, it's a 5 carbon containing one, isn't it? So 5 carbon containing one is nothing but a isoprene unit. Okay, so I will maybe what I will so this is pyrophosphate isoprenyl pyrophosphate moiety is obtained. So this on isomerization. Isomerization means this bond pair is come over here and this picks up this proton. A isoprenyl pyrophosphate is obtained. So then afterwards, if there is a combination of two isoprenyl pyrophosphate, it is possible to get a geranyl pyrophosphate. So your geranyl pyrophosphate and neryl pyrophosphate, just the difference in the cis trans isomerism. If there is a trans, it's a geranial. If it is a cis, it's a neryl pyrophosphate. And if it is a neryl pyrophosphate, this double bond means pi bond can react with this carbon and this will go out. So that is how a positive charge is formed on this tertiary carbon atom. So this was attacked by water molecule and that is how a alpha terpineol is obtained. Okay, this is about how the alpha terpineol is synthesized in a biosynthetic manner. Okay, so just I will repeat the explanation. So mevalonic acid, it undergoes phosphorylation with ATP. Obtained one is monophosphate. The monophosphate again treated with ATP gives diphosphate. This diphosphate undergoes dehydration and decarboxylation, giving a intermediate of isopropyl pyrophosphate. This combination of these two isopropyl pyrophosphate forming a geranyl pyrophosphate and neryl pyrophosphate. So this neryl pyrophosphate, the bonded pair present over here can form a ring structure and pyrophosphoric oxide will go out as a byproduct, forming a carbocation. This carbocation is attacked by water and that is how the alpha terpineol is obtained. So with this, I will go to the next slide. So next slide, how this alpha terpenol is converted into alpha-pinene, how this alpha terpenol is converted into fenchone. So you know that this bonded pair can form a bond, okay, with this carbocation. So that means there will be a new bond formation between this carbon and this carbon. So thereby leads to a formation of four-member bicyclic compound. One is four-membered ring. So another one is one, two, three, four, five. Okay, another is five-membered ring. So that is how a bicyclic compound is obtained. So this bicyclic compound, if there is a loss of proton, okay, that means this bond pair can come over here and the proton can be go out. So that is how a alpha-pinene is obtained. Similarly, suppose if this carbocation, I think this Wagner-Merwin rearrangement, this we have discussed in our earlier camphor synthesis, while the method of preparation, commercial method of synthesis of camphor. So you know that this is a thermodynamical cation. So this can be converted into a kinetically favored cation. So that means a four-membered ring is gets converted into five-membered ring. So that is what obtained over here. So this on treating with water, so what will happen? There will be a new bond formation between this oxygen and this carbon. So that is how a borneol is obtained. This borneol undergoes oxidation. Okay, I know that you know that in biological system, oxidizing agent is NAD plus. So NAD plus means nicotinamide adenosine diphosphate is the NAD plus. So this NAD plus picks up a hydrogen H2 and converted into NADH. So that means it takes H minus, okay, convert it as a NADH. So you know that NADH is acts as a source of hydride source. So that will act as a reducing agent. NAD plus will act as a oxidizing agent because it takes two electron plus proton. So H minus, it will take H minus. So that means the H minus present over here, so that will be going over and the bond pair form a double bond here. So that is how a fenchone is obtained. I think you are aware about the application of NAD plus in biochemical transformation. So one unit was there in your third semester, no? So I think you are familiar with this. So this its role is conversion of its role is oxidizing agent in a living system. It converts alcohol into uh, ketone. So similarly, right, okay, that is how a fenchone is obtained. So with this, I will go to the next slide. So I am talking about the how the biosynthesis of linalool. Okay, I think you know that this is a, neryl pyrophosphate, and this one is geranyl pyrophosphate. So neryl pyrophosphate or geranyl pyrophosphate. Suppose this OPP means pyrophosphoric acid is going out as a living group, then there will be a positive charge on this carbon, then there is a positive charge on this carbon. Okay, so what will happen if there is a positive charge on this carbon? This bonded pair can come over here. Why it is coming? Because there is a positive charge on primary carbon atom. When this bond pair is coming over here, so there will be a formation of positive charge on the tertiary carbon atom. So formation of tertiary carbocation is more feasible, and tertiary carbocation is stabilized, more stabilized through resonance. The resonance may be a hyperconjugation one, okay, and compared to the primary one. So that is the reason this intermediate is obtained. Similarly, geraniol pyrophosphate OPP, when it is going OPP as a living group, forming a cation here. So once the cation is formed, this bond pair can come over here and forming a same intermediate which was obtained from the neryl pyrophosphate. So this common intermediate, when it is again, when it is treated with the OPP, okay, so then there will be a formation of compound like this. This OPP on hydrolysis, okay, giving rise to linalool. Okay, this is how the linalool is obtained biosynthetically. Okay, just I will repeat the explanation of biosynthesis of linalool. So, biosynthesis of linalool can be obtained from neryl pyrophosphate or geranyl pyrophosphate. So, how these two are obtained? These two are gives a common intermediate called a tertiary carbocation, and after the formation of tertiary carbocation, the OPP can attack again, forming an intermediate. So this on hydrolysis giving rise to linalool. Okay, this is about the synthesis, more particularly biosynthesis of linalool. So with this, I will go to the next slide. So next slide is about mm, biosynthesis of farnesol. Okay, so here what you can observe, geraniol pyrophosphate when it is treated with isoprenyl pyrophosphate. So geraniol pyrophosphate on reaction with isoprenyl pyrophosphate. How this will react? This bond pair can come over here, and this bond pair will form a new bond with this carbon, and OPP will go out. Here the living group is OPP. So this part, okay, the CH double bond CH2, CH, this CH is this, okay, CH is C is this, CH3 is this, these two carbon atom is this, and these two carbon atom is this, methyls are this, these methyls are here. And now there will be a new bond formation between this carbon and this CH2. So that means what is happening here? You can observe that this is one saturated carbon and this is also saturated carbon. So that is how you can say that this is one, this is one more. Okay. And afterwards you can found a double bond and then CH3. Okay. This is the double bond and this will be the CH3. Then afterwards, CH2 is this is the CH2, and this will be the OPP, pyrophosphate. Then what is the name of this compound? This compound name is farnesyl pyrophosphate. Okay, this is the farnesyl pyrophosphate. This farnesyl pyrophosphate on hydrolysis gives farnesol. So this is how if somebody, I think somebody is waiting, I will enter it.
Sir, PPT
Yes,
slide.
1 minute, 1 minute. Yes, any doubt? Nobody is waiting. Okay,
Sir,
tell me.
PPT slide changing.
PPT?
Slide is not changing.
1 minute. I will. So I think so it's a farnesol. So I'm talking about the farnesol. Could you see the farnesol biosynthesis pathway?
Yes, sir.
Oh, fine. Then no problem. Okay. Maybe previous one, linalool. Now I change it to linalool, biosynthesis of linalool. Hello. Hello. Fenchone. Any doubt? Hello.
No, sir.
Okay, fine. Uh, is anybody is waiting? Okay. So now, okay. Could you see the slide biosynthesis of farnesol?
Yes, sir.
Okay, fine. So that means the farnesol is obtained by the combination of, I will take a pointer. Right. So farnesol is obtained by the combination of geranyl pyrophosphate and isoprenyl pyrophosphate. A intermediate called farnesyl pyrophosphate is obtained. So this farnesyl pyrophosphate and hydrolysis giving rise to farnesol. So this is how the farnesol is obtained from the geranyl pyrophosphate. So with this, I will go to the next slide. So next slide is about the biosynthetic formation or synthesis of beta edesmol. So here, for this, the rearrangement, okay, the rearrangement of farnesyl pyrophosphate. So you know that this is the farnesyl pyrophosphate. So this farnesyl pyrophosphate, when the OPP is going out as a living group, there will be a formation of carbocation called farnesyl cation. So farnesyl carbocation. So this farnesyl carbocation uh, form a bond. How the double bond present over here? No, this double bond can form a bond with this carbocation and a forming an intermediate like this. So you know that this is the primary carbocation and this carbon is the a tertiary carbocation. So this may be the stable compared to this one. Then what will happen? So the water moiety so can attack this carbocation and forming a alcohol formation at this carbon atom. Then afterwards what will happen? There will be a new bond formation between this carbon and this carbon. Okay, there will be a new bond formation between this carbon and then this carbon. So that is how a intermediate is obtained like this. So loss of proton, okay, loss of proton leads to the formation of beta edesmol. Okay, this is how the beta edesmol is obtained, starting with farnesyl pyrophosphate. So it's only the rearranges. Farnesyl pyrophosphate rearranges. How it is rearranges? Just by the loss of OPP, a carbocation is formed, and there will be a new bond formation between this carbon and this carbon, cyclization, and afterwards a water is attached to this carbocation forming an alcohol. So later on intracyclization. So that means there will be a new bond formation between this carbon and this carbon. So this will picks up a proton. So that is how a carbocation is obtained. This carbocation on when it is undergoing a loss of proton, forming a beta edesmol. So this is how a beta edesmol is obtained. Okay, so with this I will go to the next slide. So next slide is about how the abietic acid is obtained biosynthetically. So you know that this is about the farnesyl pyrophosphate. So here the farnesyl pyrophosphate is written in a linear form. Okay, this is the linear form of farnesyl pyrophosphate. linear form of farnesyl pyrophosphate, when it is treated with isoprenyl pyrophosphate. Okay, how the bond formation? There will be a you know that the bond pair, okay, can come over here, and this bond pair can form a bond with this carbon atom. So here, what is the leaving group? OPP is the leaving group. So that means there will be a new bond formation between this carbon and this carbon. So let's look into that. CH is this, CH3 is this CH3 is this, double bond is this, CH2 is this, CH2 is this, CH3, CH3, double bond, two CH2s, two CH2, CH3, CH3, double bond. Here, one CH2 is there, no? That is this. Now there is a new bond formation between this carbon and this carbon. So that means this carbon is this, and then this carbon is this CH3 is this, and newly formed a double bond, and then this CH2 is this OPP. Okay, so this is how the product is obtained by the combination of a farnesyl pyrophosphate and isoprenyl pyrophosphate. So this linear structure is rewritten as like this. Okay, so here just I am showing how the naphthalene moiety is formed with this linear structure. Okay, these two CH2s are here, this double bond is here, and then these two CH2s are these two, this is one, this is another, and this double bond CH3, double bond CH3, and these two CH2s are here, and then double bond. Okay, double bond CH3 is this, and then two CH2s, these two CH2s, this double bond CH3 is this, and CH2 is this, and OPP is this. So a linear structure one is converted into this form. So at this stage, what will happen? And you know that in living system, base part of the enzyme, acid part of the enzymes are present. So that means certain enzymes, more particularly a glutaric acid, or otherwise acidic amino acid containing enzymes can act as a source of proton. Similarly, basic amino acids containing enzymes can act as a base. So what will happen? The base part of the enzyme can picks up a proton. Okay, that time what will happen? The bonded pair is moves over here. Uh, okay, then this bond pair, there will be a new bond formation between this carbon and this carbon. So that is how one six-membered ring is obtained. Similarly, there will be a new bond formation between this carbon and this carbon. So that is how a naphthalene means this is a saturated, no? So we have to say that decaline. So how the decaline is obtained. And the remaining part, that is CH2, 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 CH2. And this double bond CH3. This is the double bond. This is the CH3. And since there is a loss of proton here, there will be a exocyclic double bond. So that is this is a exocyclic double bond. And after this double bond, CH2 is there, that is CH2, OPP is there. So that is how the transformation of this moiety into this one. The obtained one is called labdadenyl pyrophosphate. Okay, this labdadenyl pyrophosphate again undergo cyclization. How the cyclization? There will be a one more six-membered ring formation here. You can observe that this is one carbon, second carbon, third carbon, fourth carbon, fifth carbon, and then sixth carbon. So that means there will be a new bond formation between this carbon and this carbon, and this double bond moves over here, and this OPP will go out. So that means here there will be a living group is OPP. Then what we can explain here, so that means there will be a formation of vinyl group. This is a double bond means vinyl group, that is vinyl group. And one this CH3 is there, no, that is this one. So here when I'm writing from here to here, I will make one group as a away from the observer, another group as a towards the observer. So small CH3 one is towards the observer, bulky vinyl group is away from the observer. So that is how the product is obtained from, that is how the product formed from this moiety. So now what I will we will discuss now. So since there is a involvement of this bond pair in a new bond formation, this carbon attains a positive charge. That's what I have written here. So this carbon is a positive charge. At this moment, the bond pair, okay, this bond pair will come over here. So what it means? It means that there is a loss of proton. So when there is a loss of proton, there will be a double bond formation. That's what obtained here. Once there is a proton formation here, this proton can be obstructed by this double bond. Means this pi electron can obstruct a proton, converted into a CH2 into CH3. That's what I have written here. So once there is a new bond formation between this carbon and this proton, this particular carbon, CH2 carbon, attains a positive charge. So now you know that emigration, okay? So the CH3 groups present over here, it will migrate from this carbon to this carbon. So what it means, this is, you know that this is a primary carbocation, and this will be the what is called tertiary carbocation. This you may consider it as a Wagner-Merwin rearrangement. So if there is a possibility of forming a stable carbocation, then that migration is called Wagner-Merwin rearrangement. So that means a primary carbocation, is it a primary or secondary? Secondary carbocation. So this CH3 is moved from here to here. Then afterwards, the formed one is a tertiary carbocation. So this you can consider it as a Wagner-Merwin rearrangement. So at this moment, what you can uh, think about, you know that this proton is called allylic proton. So since there is a allylic, means allylic protons are acidic in nature. So that is the reason this bond pair can come over here. This bond pair will move over here. So that means there is a loss of proton. So that is how a intermediate formed like this. So from here to here, so it is possible to get abietic acid. How? What is the difference between this and this? Only the here the CH3 group is there. Um, this CH3 group is converted into COOH group. Okay, so that means addition of oxygen. Okay, addition of oxygen means there should be a involvement of oxygen, oxidation. Okay, I think maybe you are in biochemical mechanism you learned about the addition of oxygen, oxidation, reductions. So this may require 2 or 3 steps. The CH3 is gets converted into COOH. More particularly, maybe the involvement of cyanocobalamin, it is possible to transfer CH3 group into COOH. So that is how the abiotic acid is obtained biosynthetically. Okay, what I will do at this moment, just I will explain about this repeat the explanation about the biosynthesis of abiotic acid. So this is farnesyl pyrophosphate. This is isoprenyl pyrophosphate. When both are combined, it gives a intermediate like this. This intermediate we can represent it in a different form. Okay. What will happen here? A cyclization. Cyclization forming a decalin moiety. How the cyclization? There will be a new bond formation between this carbon and this carbon. So that is how a six-membered ring is formed. And there will be a new bond formation between this carbon and this carbon. That is how one more six-membered. Both the rings six-membered. Now it's called a transdecalin derivative. So this particular name of this derivative is called lambda-dinyl pyrophosphate. So this again undergoes cyclization. How there is a possibility of forming a six-membered one more ring? It's called this. Now what you can say that one, two, three these three rings. Okay, how you can get this? May be a kind of uh, what you call saturated phenanthrene skeleton 1. Okay, and now since there is a positive charge, this bond pair going over here, so this proton was captured by vinyl pi electron, forming an intermediate like this. And here there will be involvement of Wagner-Merwin rearrangement, because there is a migration of 1,2 shift. Okay, 1,2 shift of methyl group, and the obtained one, since there is a allylic proton movement of loss of allylic proton, the bonded pair can come over here, this bond pair can come over here, forming a intermediate. This intermediate can be converted into abiotic acid, maybe in the one or two or three steps, maybe the involvement of oxidation process. Okay, it may also involvement of the cyanocobalt amine. Okay, that is how a abiotic acid is obtained. So this is about abiotic acid. Okay, maybe what I will do, uh, uh, maybe I will can go for this. Okay, maybe I will stop at this stage. Maybe what I will do now, I will share the questions. Okay, so maybe what you can do, you take the screenshot or the copy the questions, and after copying a questions, you have to answer those questions in a blue book or a notebook. But blue book or notebook, that you have to submit when you are coming to the college, and you have to write this in presence of your parent. Take your parent signature, then afterwards make one PDF file and send it to mail.
Sir,
Okay.
Blue Book is not available, sir. No shops are open.
Fine. Then you maybe you tear some
We'll
pages
write
of
in
your.
some pages, sir. We'll write in
No
pages.
problem. No problem. But you had to submit that copy, that you had to remember. And
Yes,
one
sir.
more
We'll
thing,
submit.
you have to make a PDF file and send it to my mail. Okay. Okay. Now I will share the question paper.
Okay, sir.
Okay, 1 minute. You be in the line. I will tell you. Okay, I think now you can see the slide, no? More particularly the Department of Chemistry, MSc Chemistry, fourth semester, first internal CH 403, Industrial Chemistry, Heterocyclic Chemistry. I think visible, no? I will put it in a presentation mode. Is it okay?
Sir, nothing is visible, sir.
No, I think you can see that it's visible.
For
No, sir, it's not visible.
No, sir, it's not
It's not
visible,
visible.
sir.
If that is the case, I will stop sharing and again I will do it.
okay, sir.
Is it visible now?
No, sir. Now it is visible, sir.
No. Okay, may note down it. Maybe you can copy it. I will remove later. So if you have any doubt in the question, so you can also ask me.
9 questions, right, sir?
Yes, it's 9 only. It's one slide. And here up to fifth question, it's carries 2 marks one, and the remaining ones are carries 3 marks.
Okay, sir.
And there is no choice. All the questions you have to answer. Copied, no?
We took screenshot, sir.
Then I can stop sharing, no?
Yes, sir.
Hello?
Yes, sir.
Okay, that fine. I will do it. That's all about today's class. Maybe tomorrow, as usual, I will take a class. Okay. Now I will